All, all right, it's 7 o'clock, so let's get started. Good evening. Welcome to the environmental lecture presented by the American Chemical Society Pittsburgh local section. My name is Ron Hong Lin, and I'm a member of the ACS Pittsburgh local section executive board and chair of the environmental group. The ACS Pittsburgh local section is dedicated to building a vibrant and sustainable community through the power of chemistry. The environmental group is one topical group within the local section formed with the aim of educating and promoting awareness about our environment. We support a variety of activities. Tonight's lecture is the third of the environmental lecture series, which we plan to continue monthly throughout the year. We intend to continue with a variety of speakers from academia, industry, as well as other research institutes, and cover various chemistry and environmental science related topics. Just to let you know that the lecture is being recorded and will be posted on YouTube later. I'd like to remind the attendees and future viewers that the views and opinion expressed in this and any future lectures are solely those of the presenters and do not necessarily represent the views of the ACS Pittsburgh local section. Our next lecture will be on Wednesday, July the 5th at 7 p.m. featuring Professor Wenchai Zhang from the Virginia Tech. And he will be discussing rare earth element recovery from coal-based materials, progress from feasibility assessment to pilot scale RE production. Now, it's my great pleasure to introduce tonight's speaker, Dr. Carol Ng. Dr. Ng is an associate professor in the Department of Civil and Environmental Engineering at the University of Pittsburgh, with secondary appointments in environmental and occupational health and in chemical and petroleum engineering. The research in her group focuses on developing models to understand where chemicals go in organisms and ecosystems using tools from chemistry, biology, and engineering. Active research areas include modeling the accumulation of the forever chemicals, or the PFAS, in wildlife and humans, checking the complex chemical mixtures in the environment, and exploring the role of the industrial food system on human exposure to persistent and toxic chemicals. Dr. Ng received her BS and MS degree both in chemical engineering from the SUNY Buffalo and her PhD degree in biological engineering from Northwestern University. With that, please join me and welcome Dr. Carol Ng, and thank everyone for joining tonight's lecture.
Thank you so much, Rong Hong, for the invitation to speak with you all tonight. And let me get my slides shared here so that we can get going. Okay, so hopefully you all can see this okay now.
Yeah.
Yes, so thanks so much for having me, and hopefully this will be of interest to your community. And it's my understanding that you've recently had another PFAS lecture. And so I'm not going to go very deeply into defining PFAS, but I think it's always helpful when you have this class of chemicals that can turn into a sort of acronym soup to just give you a little bit of guidance as I go through the slides today to kind of give you an idea of how to orient yourself around PFAS. So the two most well-known, let me get my little laser pointer going here. The two most well-known PFAS that are the most well-studied are PFOA and PFOS. These are what we call legacy compounds because they are no longer being actively used, at least in the US, Europe, and other countries. There are some places where they're still being used. And these are examples of what are known as perfluorinated substances. So these are organic compounds where all of the carbon-hydrogen bonds, except for the ones in the acidic head group, have been replaced with carbon-fluorine bonds. And they're just one type of the many types of PFAS that are out there. And they're most famous for being highly surface active compounds that make excellent both stain and water repellents. And that led to their widespread use in lots of consumer products like the well-known Scotchgard and Teflon applications. In addition to that, there are many other PFAS. So for example, there are partially fluorinated or polyfluoroalkyl substances. For example, this fluorotelmer alcohol I have on the right here. This is an 8,2 FTOH because it has 8 perfluorinated carbons and 2 carbon-hydrogen bonds. And compounds like this FTOH are sometimes referred to as precursor compounds because due to the fact that they have some breakable bonds, they can transform in the environment. And these transformations can lead to the formation of short-chain PFAS and to also some ill-defined transformation products. So there's also a lot of sort of PFAS dark matter where some more complex chemicals can react to form them. So because especially of the surfactant nature and very unique properties of PFAS, they have come to be used in a dizzying number of applications. So this includes applications in applications in safety. So one very well-known one that has also led to widespread environmental contamination is the use of PFAS in so-called AFFF or aqueous film forming foam. These are firefighting foams that are intended to be used for things like hydrocarbon fuel fires, so fires that have a fuel source that are very difficult to put out. PFAS are excellent at creating a smothering blanket on top of such fires and puts them out quickly. In addition to firefighting foams, there are a lot of occupational health and safety applications for PFAS. So one important example is in surgical gowns for long surgeries because PFAS make these gowns both resistant to the penetration of biological fluids while also being breathable for long wear. In addition to these sort of specialized uses, there's also lots of consumer-facing uses, including the use of PFAS in food packaging. As I mentioned previously, these chemicals are special in that they are both water and oil resistant, and people want to avoid the leaking of oil through especially paper-based food packaging. And so things like pizza boxes, burger wrappers, and cookie wrappers are often impregnated with PFAS in order to impart that oil repellency. Surprisingly, they're used in a wide range of personal care products, and this is the one that often sticks out to people that they didn't realize that PFAS were used. So for example, certain brands of dental floss that glide quite smoothly between your teeth, they glide smoothly because they include Teflon. And Teflon is a fluoropolymer, so it is sometimes argued that it itself is not a PFAS because it's a polymeric material and it's not a free molecule, but there can be PFAS still associated with fluoropolymers, and they're certainly associated both with their manufacture and with their end of life. And PFAS are also used in things like shaving cream and even waterproof makeup. So there's a lot of uses where people are directly exposed to these compounds. And importantly, as we move into some really important future technological applications like the manufacture of semiconductors and the use of smartphones and other surfaces that need a stain-resistant coating, like this oleophobic coating that is meant to be put on the screen of your cell phone to prevent fingerprints, there are many uses of technological uses of PFAS that are also important. And finally, there's a lot of uses of PFAS in pharmaceuticals. Now, these tend to be a lower degree of fluorination than you see in all of these other products, and it's often the addition of 1 or 2 fluorinated moieties onto an active drug compound, which makes it both stick around longer and break down, be resistant to breakdown in the body, which can extend the active life of a pharmaceutical compound. Now, this list that I have here is illustrative, but not at all exhaustive. I've been involved in publications trying to understand the uses of PFAS that have identified more than 1,000 compounds in more than 200 different use categories. And so these compounds really have pervaded all aspects of modern life. Because of this, PFAS are now found everywhere. Everywhere we look, we find them. And that refers both to the environment and, unfortunately, to our own bodies. So there are recent studies that have shown that PFAS are now detected in rainwater in parts of the US. And because, unfortunately, many of PFAS are now known also to be toxic, there is concern that the levels that are found in rainwater are now beginning to exceed the health advisory levels that have been proposed for some of these compounds. They are also found in the most remote environments. So here's a paper from 2020 that looked at the presence of different PFAS in the Arctic. And what's interesting about this is that, if you look at what we think of as legacy compounds, compounds that have been used for a long time, like PFOA, the Arctic is now a net source of these compounds because they've been accumulating there for so long. And at the same time, they are receiving newer compounds like Gen X, this hexafluoropropylene oxide dimer acid is the technical acronym for the compound better known as Gen X, which is an emerging PFAS. And sadly, PFAS are now found in the blood of almost everyone on Earth who has been tested for it. And not only do they get into our blood, but there are studies that show that PFAS can cross important biological barriers. And that includes the blood-brain barrier, so they can get into our brain. And it also includes barriers like the placenta and cord blood. So since we know we all have PFAS in our bodies, how does it get there primarily? If you're a person who lives near a contaminated site, excuse me, then drinking water is usually the major source of exposure. So if you live near an airport, if you live near a military facility where they've had a lot of use of this AFFF, it's likely that drinking water is your primary exposure route. However, if you don't, and we're lucky that in most places here around Pittsburgh, our drinking water tends to be fairly low in concentration for most PFAS, then probably without major drinking water contamination, food is the major source of exposure. And I just want to note this little caveat here that almost all water, like I showed earlier, even rainwater has some PFAS. Contaminated sites will have much higher levels, which is why that switches over to your major exposure route. And then finally, there's a lot of ongoing research to better understand other sources of exposure to PFAS, including personal care products, cleaning products, carpets, and air, both indoor and outdoor. So knowing that we have this massive problem out there, there's been a lot of attention paid to PFAS. And there's a lot of people working on useful ways that we can describe PFAS and predict where they may show up next. And because my training is in chemical engineering, and I mostly did modeling work coming up as a graduate student and even into my postdoctoral period, I approached the problem of PFAS the way that I've been approaching the problems of other compounds that I had studied. So for example, when I did my PhD, the focus of my work there was on PCBs, which is a very well-studied persistent organic pollutant. And for compounds like PCBs, like DDTs, like a lot of flame retardants, there's a class of models called multimedia box models that have been extremely useful in environmental science and engineering. And they're a very nice way to both formalize and simplify how we can describe where chemicals go in the environment. And they've been actually very useful, especially for persistent chemicals where global distribution becomes an issue. There was a nice paper in 2010 by Matt McLeod, Tom McCone, and Konrad Hungerbuehler. Konrad was my postdoctoral advisor, that talked about, whether mass balance-based multimedia models were dinosaurs or if they could still be used. And part of the reason for that is there was an emergence of certain compounds for which these approaches weren't working as well as they had been in the past. So for those of you who are not familiar with multimedia mass balance models, they basically take an entire complex environment and they break it up into a series of interacting boxes. And so you have a box for the atmosphere, a box for soil, a box for water, a box for vegetation. And then there are these different rates and processes of exchange of chemicals, across these different environments. And these have been used in the past to look at the global distribution of DDT, the global distribution of PCBs. They're great because they can model things that are quite volatile and will be mostly in the atmosphere, but also things that are semi-volatile, which are some of our most important persistent organic pollutants that have this dynamic relationship between partitioning to aerosol particles versus being in the air versus depositing to water, et cetera. So these models are fairly powerful. Their framework includes chemical inflows and outflows, transformations and partitioning, and they're well established for a lot of persistent organic pollutants at multiple scales, and they've even been used in a lot of contexts for regulatory screening, so they are accepted by regulatory bodies. However, they do assume that there's homogeneity within the boxes. This is what allows them to be quite simplified. And because of this, and because of the way that they generally operate, they really rely on what are called equilibrium partition coefficients. So equilibrium partition coefficients, things that you probably remember from your chemistry classes, are like the Henry's law constant that allows you to understand how a chemical transfers between water and air. And also the very famous octanol water partition coefficient, which has been the basis for understanding aerosol partitioning, soil partitioning, water sediment partitioning, and even biological partitioning. So the octanol water partition coefficient has basically driven a ton of our understanding of how chemicals behave in the environment and in organisms. Unfortunately, this very well established, often used model of the octanol water partition coefficient, which is KOW, sometimes also referred to as log P for the log of the partition coefficient, does not work with PFAS. So the way that the KOW works is by assuming that the distribution of a chemical in an organism is largely driven by its accumulation in lipid phases. But PFAS, unlike almost every other persistent organic pollutant we know, do not accumulate in structural lipids. They don't find them in adipose, right? So, if you've ever been a person who likes to go fishing, there's a lot of fish advisories out there, and they'll give you advice that says, for example, you should only eat one fish a week for what you catch from this water body, or once a month. And if you do eat the fish that you catch, you should remove the skin, you should grill it, remove the fat, and that'll reduce your exposure. Sadly, that doesn't work for PFAS. When we try to model PFAS using this KOW approach, if you look at the left-hand plot here, this is looking at the bioaccumulation, the bioconcentration factor. So the ratio of the chemical compound found in the organism divided by the amount found in the water versus the predicted log BCF, we find that if you only use partitioning, that's these X's here, you way underestimate the amount of chemical that builds up in an organism. And in fact, that these are much more bioaccumulative than you would expect, given that they don't build up in the fat. And so we had to come up with better ways to model how PFAS accumulate in organisms, because the discovery of PFAS in almost everybody's blood was a wake-up call that these chemicals are not only persistent, unfortunately toxic, but also very highly bioavailable. So how could we modify our understanding of how these behave in biological systems? So I had come from a background of using these partition coefficients, and I was looking for a better way to try to understand how PFAS accumulate. And so as I was researching into how people model biological systems, I started studying a different class of organism accumulation models, and these are PBPK models. So independently from the development of these multimedia partitioning-based models, there's been a long history of building up what are called toxicokinetic models to model chemical concentrations in biological systems. And toxicokinetic models are much more common in mammalian toxicology, in human toxicology, and had been much less so on the ecotox side, which is kind of where I came from, because they're much more complex. So these began their development with data-led empirical models, where basically they would run experiments in vivo. So they would expose an organism to some contaminant, and they would collect data on how much was accumulating and then being eliminated from different tissues. And then based on these data, they would come up with a fairly complex description of the different tissues in an organism and come up with a model that would describe the disposition of the chemical in these different boxes. And so where the multimedia mass balance model started from first principles and worked towards a prediction, this is an empirically led model that started from the data, and then from that derived the model structure. And so what I wanted to do is kind of figure out where these two models meet. So if you think about the different levels of model complexity, the partitioning-based KOW-driven lipid partitioning model is on the very simple side. You can get the next level up of a toxicokinetic model that is just a one box PBPK model that either models blood or not blood. So there's, the main part where the PFAS accumulates and the rest of the body. This is also sometimes referred to as the volume of distribution. And then you can get more and more complex by including other compartments, other tissues of interest, and the rest of the body. And then you could even get more and more complex, and I'll show you why we kind of needed to go in that direction for PFAS, and not only look at different tissues, but also what is the composition of those different tissues, what are the specific cell types, and how might those specific cell types react with the chemicals of interest. And then within the different cells, what are the specific proteins? So this is how we start out with what's I sometimes refer to as a bag of lipid model and go all the way to the molecular level model where we're interested in these very specific interactions of PFAS. So why did we need a model this complex to be able to capture the behavior of PFAS? Well, there are a lot of key observations that show that they're very different from your typical persistent organic pollutant. So at the top here, we have one particular study that looked at the preferential accumulation of PFAS in different tissues. So if you look at the PFOA level, this is the tissue to blood ratio. It shows that the liver is by far the highest, and then everything else is really quite low. And if you look at also the differences across different species, we find that PFAS behave very differently, not only according to the different types of PFAS. So this is organized by the chain length, getting from the shortest chain PFAS to the longest chain PFAS among these, but also across different species and even within a single species between the male and the female. So the y-axis here is the PFAS elimination half-life in days. So it tells you a little bit about the elimination rate or how long it sticks around in the body. I'd like you to note that this is on a log scale. So it goes from hours to years, essentially. And you can see that, for example, for PFOA, the half-life of PFOA in the female rat is only hours, but it's days in the male rat, and in the human, it's years. There's something to do with body size scaling here, but not all of this difference can be explained by that, because even within relatively similar species, so here you have the rat, the mouse, oh, and I don't have it here anymore. I took the rabbit out. Even within similar rodents, the half-life differences can be quite extreme. So one of the reasons that I started thinking about why this could be was due to specific behaviors of PFAS that mean that it interacts more strongly with biological receptors than other chemicals have in the past. And the key to me for making these connections and analogies was to look at the similarity between this very famous PFAS, PFOA, and something that's really important to our body, which is a fatty acid. So on the left, we have perfluorooctanoic acid, where all of these carbons are bonded to fluorines. And on the right, you have octanoic acid, a saturated straight chain fatty acid. And we have a lot of machinery in our bodies that are specifically evolved and targeted to process fatty acids, right? Fatty acids are a building block of cells and of amino acids and all sorts of things in our body, and lipids. So our body has already in them many fatty acid carriers that are meant to transport these hydrophobic substances around the body. And two key ones are serum albumin. This is one of the major proteins in our blood. And another one is the fatty acid binding protein, of which there are several forms. This is one of them. This is the liver type fatty acid binding protein. And the function of these proteins is to take a fairly insoluble compound and shuttle it around either within the body through the blood or inside of a tissue in the cells from the cell membrane to the organelles of the cell that will then process that compound into other things. In addition to these binding proteins, there are also proteins that are located in the cell membrane that help these hydrophobic substances that have, these polar ends be able to pass through the cell membrane. And so there are organic anion transport proteins and polypeptides that will shuttle these compounds back and forth. And an interesting thing that may help explain this very large difference between the male rat and the female rat in their half-life is that some of these transporters are preferentially expressed in one sex or another, and they can be responsible not only for removing compounds from an organ, but also reabsorption of organic anions. So there are organic anion transporters in the kidneys that reabsorb PFAS from the blood, thinking that these PFAS are actually fatty acids that we need, right, for our nutrition. And so they've proven that if you treat rats with, if you treat male rats with estrogen, suddenly their half-life will drop to match that of the female rat. So they can show that it's related to these sex-dependent protein expression. So that's a lot of information, but this is sort of how we build the case for what is it that is leading to some of the unique behaviors of PFAS inside the body. And so when I started my position at University of Pittsburgh in 2016, I was lucky enough that one of my first students was a really brilliant person. His name is Weishao Cheng. And he took a model that I had started working on as a postdoc that looked at PFAS in the rainbow trout. And he modified that model to describe PFOA in the male rat. And he chose the male rat because it's one of the organisms that has the most physiological data available. And he really built this PBPK model from the ground up by describing all of the physiological parameters he could find for some of these key tissues, and then finding as much information as he could about all the proteins in the rat body that might have important interactions with PFOA based on their interactions with fatty acids or with established data on actual binding to PFAS. So we looked at observations of accumulation and elimination. We looked at specific proteins in the blood, the liver, and the kidney, and found as much both in vitro and in vivo data as we could. So this led to a model that could include protein binding, transporter interactions, and really was able to successfully explain observations. So with this model that was not fitted to any data, so it's different than old approaches to BBPK, he was able to successfully predict in the black lines here the colored symbols, which are the measured data, that was looking at PFAS exposures at different doses and both through oral and IV routes. So it had a really successful model that could look at tissue distribution, and was also successful for the time course. So if we look here, this is the concentration over time in the blood as a result of IV dose at high concentrations, oral dose at high concentrations, IV dose at low concentrations, and oral dose at low concentrations. And we were able to calibrate the model by using a Bayesian approach that really helped us tighten these uncertainty ranges, which, of course, in order to do this, you need really good experimental data. So we had two data sets that kind of disagreed, and one data set was bigger than the other. And so our calibration really pushed our model towards that larger data set, which was an older one. So again, if you need to calibrate your models, that kind of leads you toward really needing good data. But this was a massive exercise that took a ton of data collection. It was really only possible for the one chemical for which we had a lot of information. And so one thing that we were left thinking about is that, you know, this type of approach using a PBPK or PBTK toxicokinetic model to describe PFAS in an organism, much more successful than a partitioning-based approach, because it can integrate understanding of uptake, internal interactions, and these elimination processes and how they can vary with different proteins. But it takes so much data to parameterize a model like this that it really impacts its generality. So this is, you know, we made a model for PFOA because most of our information is about PFOA and PFAS. That's where most of the publications are out. And so this brings to mind this old parable about, you know, the blind men trying to describe an elephant. This is basically where we are. We know, all we know, or the majority of what we know about PFAS are about these two front toenails, right? And there are so many thousands of compounds out there for which we have very little data. So what kind of forest are we missing because we are only focusing on these two trees? So we started thinking about what information do we need in order to make these models more generalizable, and how can we get that information without hundreds of years of more research compound by compound basis. So the first thing we have to say is that our understanding of the interactions of PFAS with these biological systems is still incomplete. So the way that we were able to calibrate our model, we used a Bayesian approach that took our prior distributions for all of the different physiological and protein interaction parameters, and then we calibrated it to available distribution data so that we could then update our parameter distributions. That's the posterior distribution shown here. And while some of our knowledge was pretty good, you see very little difference between the prior distribution and the posterior distribution, there were some that were obviously way off, right? So we ended up with more uncertainty about the renal clearance rates than we started with. And it turned out that the blood is much more, sorry, the blood membrane transfer is much faster than we would have predicted. So the effective permeability of the blood compartment is much higher than we originally thought. So this is an empirical stand-in for partitioning, and we also don't understand enough about transport and binding. So what can we do to improve our understanding here? So PFAS taught us that bioaccumulation happens differently from tissue partitioning. And we need to be able to identify tissue-specific components in order to really understand the differences across different PFAS. So here is a really interesting paper from Clifton Dassam-Cow. He came from Elsie Sunderland's lab at Harvard that was looking at the distribution of different types of PFAS with different chain lengths in pilot whales. And he saw that not only liver and blood were really important, but also the brain. And in some cases, the brain was even more important, like, for example, for this long chain C14 PFAS, than the liver in accumulation. And with this, he also looked at the correlation between the total protein content in those tissues and the total phospholipid content in those tissues, and showed that there was a higher correlation for some tissues like the brain with the phospholipids and not with the proteins. And so there might be other tissue-specific components that are really key in determining accumulation in these different tissues. In addition, lower bioaccumulation does not necessarily mean that something is safer. So this is a paper that's from Bevan Blake, from Sue Fenton's group at the time, that was looking at the impacts of PFOA versus Gen X. As I mentioned, this was this emerging compound that took over from PFOA when PFOA was phased out. And shows here that Gen X is as toxic, but in different ways as PFOA in developing mice. And so it's not just the PFAS that bioaccumulate that we need to look into, but there also might be some specific mechanisms of toxic action that need to be paid attention to as well. And finally, there's the idea that there are a lot of PFAS precursors out there. And so this is looking at different classes of PFAS that are found in indoor dust. So looking at this indoor exposure, and you can see that the well-studied PFAS are the perfluoroalkyl acids. They're here at this 1 nanogram per gram, more or less, concentration. And you see much higher concentrations of the fluorotelomere alcohols and the PAPs, which are present in a lot of personal care products and consumer products. The reason that it's important that these two are higher is that both of these are transformable PFAS, PFAS that can break down into other compounds. The precursors are present at the highest level. And you can see the same thing here from a study from Hall et al. That looked at the dust concentration in fire stations and in homes. And again, you see very high concentration of these transformable PFAS. And so understanding what these PFAS transform into is really key to understanding indoor exposures. So there are several really important needs to be able to advance these models. The first is that we need better approximations for key phases in different tissues. That includes proteins, phospholipids, so the components of the membrane that the PFAS may be partitioning into. There's differences between different proteins. So I mentioned that PFAS don't accumulate in lipids, right? That makes them really different from other persistent organic pollutants. But it turns out that they don't accumulate in just any protein either. So muscle tissue is also very low in PFAS, even though muscle tissue is very high in protein. So it seems like structural proteins are fundamentally different from what we refer to as functional or active proteins in their ability to bind to PFAS. There's also an understanding that proteins are not static, right? They are dynamic and they react to transformations of compounds in the body. They are influenced by environmental factors, so seasonality, temperature, disease, pregnancy, all elicit changes in physiology, which then elicit changes in the distribution of lipids and proteins in the body, which may then lead to altered PFAS tissue distribution. And then finally, there's this understanding that there are key differences across different organisms, and there are key organisms that we use as models for human toxicity for a lot of compounds. And so we need to understand where this is appropriate and where the limitations are for using these model organisms to understand toxicity in humans. So one of the things that we're trying to now work towards, we spent a lot of time, I'd say like the last 6 years in my group has been about getting more and more detailed and understanding all of these different complex behaviors of PFAS. And now we're trying to see if we can go back towards simplicity again in a way by managing the complexity and trying to pull out what are the key features that are driving where PFAS go and what they do. So for example, if we look at this rat PBPK model that I discussed, it includes passive diffusion for all tissues. It includes active transport for select tissues. It includes binding to serum proteins in blood and fluids, interactions with functional proteins in the liver and kidney. And there's still other things that have not been well explored. So what is the interaction of PFAS with structural proteins? We know it's generally low for the well-studied PFAS. Is that going to be true for all of them? What is the role of fat storage? Is it the same across different PFAS classes? How do cell membrane phospholipids contribute to overall bioaccumulation behavior? Could these factors improve the accuracy of PBPK models? And if so, can we find a way to still make these models, complex models, tractable? And so we're working towards coming back to the idea of partition coefficients, but a better partition coefficient. And I put this in quotation marks because it's not a true partition coefficient. These are not systems that are at necessarily equilibrium. And we're talking about saturable partitioning, right? So saturable binding if there's a specific binding site in a protein. So instead, we're thinking about developing these biological distribution coefficients between a tissue and its fluid. That's determined by the composition of the tissue, the different fractions that are there, what kind of proteins, what kind of lipids, and the individual distribution coefficient for each fraction with a specific PFAS. So for each tissue, we think about what are the functional proteins, what are the phospholipids, what are the storage lipids, what are the structural proteins, and then we make a weighted sum over all of these components. And so a PBPK model that includes all of this would have this somewhat more complex structure where in each fluid you have these different fractions, but then we can come up with an overall factor, this K tissue fluid, to describe how PFAS builds up in there. And so in order to do this, we have to come up with what are the different fractions of the different components. So we're looking at, for example, lipidomic studies to tell us what are the different types of lipids that are in the body and how will they interact with different PFAS. So we can look at the types of membrane lipids that are in the different tissues versus storage lipids. So we know, for example, if we think back to that paper by Clifton Dasenkow that found high levels of PFAS in the brains of the pilot whales, brains are very high in membrane lipid, very low in storage lipid. And so the PFAS are binding to the membrane lipid fraction. Whereas in the fat, it's mostly storage lipid versus a little bit of membrane lipid. And so we're coming up with this composition of the different components, the distribution coefficients of the PFAS in these components, and then what are the best ways that we can get these data for different PFAS, different organisms, estimation methods for distribution coefficients, membrane water partition coefficients. This is something we're actively working on using these solid supported phospholipid bilayer in vitro assays. There are polyparameter linear free energy relationships that can be used to estimate partitioning to structural proteins. And then finally, a big focus of work in my group is to evaluate the binding of PFAS to different functional proteins using in vitro methods like equilibrium dialysis, and we're now developing SPR for this, so that we start to build an overall picture of how each fraction of each tissue contributes to partitioning. So this is for an example for a PFOA, where you can see, for example, in the liver that this is driven almost entirely by phospholipids and binding proteins. And then depending on which binding proteins are expressed in which tissues, it might then be more driven by phospholipids than proteins. And then we would need to start to build this out for the different types of PFAS. So understanding how the passive and the active interactions lead to the behavior that's observed. So we need protein binding parameters for key proteins. We need membrane transporters to understand uptake. And then we need to use this information to inform these different tissue partition coefficients. And then finally, I just want to spend a couple of minutes talking about the toxicity piece. So we talked a lot about toxicokinetics, which is just about where did the PFAS go, what tissues do they wind up in. But another branch of my group is really focused on what happens once they get there. What are the effects of the different PFAS? And so we are also using molecular methods to try to understand this using the adverse outcome pathway framework. So basically trying to understand, if you start with a specific PFAS, what are the molecular interactions they're having, like interactions with different proteins or DNA binding that then leads to a cellular response, an organ response, and eventually organism and population level responses. And so to predict the key piece here, this molecular initiating event of where PFAS interacts with some sort of protein, we were identifying different types of PFAS toxic effects, and then identifying what are the specific proteins that are associated with those specific effects, and then finding ways to now estimate the level of interaction of different PFAS with these proteins. So we're doing this using molecular modeling. So we simulate the protein ligand interaction using molecular docking, and we simulate the protein conformations using molecular dynamics. So this combination is what's called the relaxed complex scheme, because it's more efficient to only model the protein in this flexible way and then do rapid docking with all the different ligands. And this is just an example of the different proteins that we model in this way. We're looking at liver fatty acid binding proteins, serum albumin, PPAR alpha, and transthyretin. So this is just what they look like when they are allowed to move in space and wiggle around and change their conformations. And so from that, we then select representative protein ensembles. So these are snapshots of the way the proteins move in the space. And we take these different clusters of protein snapshots and we dock them to a wide suite of PFAS to look at the relationship between the free energy of binding and the chain length of, on the left here, we have the carboxylic acids, and on the right, these are the sulfonic acids. And you can see in general, this is a trend that's been acknowledged across both computational and experimental work, that the binding of PFAS to proteins like LFABP increases with increasing chain length. But what you'll see here is that if you just do docking only, which is pretty fast, you tend to overestimate binding for the long chains, whereas the experimental results, which are in purple and red, show that this kind of levels off for very long chain PFAS. And then by improving our modeling of the protein, we can get closer to the measured empirical approach. So we can use this more efficient, but also more refined molecular modeling scheme to improve our ability to predict absolute binding affinity and also the correlation between chain length and binding affinity. So that's one way we can use computational methods to get better data, but we want to validate the computational methods, and there's still very limited data available for many proteins. And so we want to be able to improve the speed at which we can do this protein screening. And for that, we are exploring the use of surface plasmon resonance. Specifically, we're looking at the ability to get both the kinetics and the binding affinity through the interaction between an immobilized protein and the test compound. So the test compound would be the PFAS, and then the protein of interest could be immobilized to the sensor chip. You would get some sort of signal from which you can extract the binding affinity. Unfortunately, PFAS are small, proteins are large, which means that in general, it's very insensitive and you need a very sensitive instrument to do this by SPR. And so we are now exploring ways to mobilize instead of the protein onto the sensor chip, finding a way to use PFAS that have certain functional groups in them so that we can actually immobilize the PFAS onto the chip and then flow the protein over it, which makes a much larger deflection, and therefore raises the sensitivity of the method. And so with some of these chemistries, we're finding out new ways to use SPR to be able to generate experimental data to validate our models with. And in the last couple of minutes, I just wanted to leave you with an actual application of models like this that could have important implications for risk assessment for humans, for example. So I talked about how In the kidneys, there's expression of certain uptake and reuptake transporters that can lead to longer half-lives of PFOA, for example. This renal reabsorption is understood to contribute to the long half-life of PFOA in humans, and it's believed to drive the sex difference observed between male and female rats. And there's a lot of transporters that have never been tested. The other problem is that protein expression is dynamic. And so when we look at protein function and PFAS half-life, is exposure to PFAS causing changes in kidney function, or are changes in kidney function causing changes in the way people take up PFAS? And when we're trying to understand epidemiological data, we need to understand whether there's reverse causation or causation happening. And so I have a postdoc who's working with me who's been looking at which PFAS have been tested in the kidneys of both humans and rats to try to understand what would be the impact of kidney injury and kidney disease in the way that PFAS are taken up in the body. And so she's done simulations using the same model I showed before, but looking at the impacts of kidney injury in the way that it changes the expression of certain transporters and picking the lowest observed value of transporter expression and modeling that through the PBPK model to look at differences in half-lives and final internal doses of PFAS. And showing that different changes in protein level leads to differences in PFOA that can either increase or decrease PFOA half-life. So depending on the type of kidney injury and what happens to the kidney function, you may wind up having populations that are especially vulnerable to accumulation of chemicals like PFOA. And a model like this can be really handy in helping you understand that and identify those populations. So I went through that really fast, hopefully just to give you guys some vignettes of what it is that we do in my group, and to show that, to tackle a class of chemicals like PFAS that are so complex, you really need a lot of different techniques and data streams to build models that are both flexible and generalizable. This is, walking that tightrope between complexity and generalizability. And the challenge is really in identifying how much detail is actually enough not to get lost in the details. And of course, this really depends on the model purpose of what are you trying to do? Are you trying to predict something very specific within a specific tissue for a specific chemical? Or are you trying to do broader risk assessment? That'll really drive the complexity of the model that you are able to put together. And before I stop, I just wanted to make sure that I acknowledge the members of my research group because they are the ones doing all the hard work. I just get to talk about the cool work that they do. And also to thank all of our funders, which includes the US EPA, the NSF, the USDA, CERTUP, and NIH, among some others. And with that, I hope we still have a little bit of time, and I'd be happy to take any questions. And if you're too shy to ask me questions, there's my email address. You can always send me an email. Thank you.
Thank you, Dr. Very interesting talk. So now we're open for questions. You can submit your question using the Q&A or just unmute yourself and speak up. So I have a question. So just go back a little bit, like you mentioned the multiple medium box model, and it's multiple scales can be used. So what that means, like multiple scales, the different box or the in terms of the molecular, so nanoscale, meter scale, or big macro scale.
Yeah, so for the multimeter box model, like the one that's shown on the screen here, when we talk about different scales, is you can run this model at a city scale, or you can do like a model for the whole world. You can do a unit world model where you're looking at global distribution. When you're talking about biological models, typically like the PBPK models, you can do that at like the tissue level or the whole organism level. So those are usually the scales we're talking about. Yeah.
So there's different parameters involved.
That's right. So if you want to do, a city, a lot of it is about how robust your assumptions are. So if you're doing, for example, a city scale model, or you're modeling a pond and the water over the pond, the assumption that the compartment is relatively homogeneous is probably a good assumption, right? Now, when you're doing a global model, if you're doing a model that represents the entire ocean, that's not such a good approach anymore. So for global circulation models, it's sometimes better to do many multiple boxes instead of a single box to represent that entire environment. And so, of course, if you go towards, some of the atmospheric modeling, some of them can be parameterized in a similar way, but they are essentially millions of boxes that are connected to one another by flows. And then you can have the changes in the parameters as you go from box to box.
Hey, do we have any other question from the audience? Okay, actually, I have quite a few questions. So, yeah, the POPs and PFAS are so different.
Mm-hmm.
So, why it's so different? Do we understand why? Because of the fluorine, those
Yeah,
the structure.
There's a couple of reasons why they're so different. So it kind of depends on what aspect of it you're talking about. So if we look at PFAS here, there's the first thing about it is that this carbon fluorine tail is very hydrophobic, right? And so this part acts a little bit more like a pop, but this part is actually quite hydrophilic. And one thing that wasn't really understood when they first started looking at PFAS is that it's a strong enough acid that these are generally 99.9 something percent ionized at environmentally relevant pH, right? So these are not neutral compounds in the environment. They're negatively charged. And that makes them very different from the typical POP, which are almost all neutral compounds. And so they have these electrostatic interactions, and also they don't accumulate in lipids because lipids are nonpolar.
Okay, I see. Okay, my next question is, so in your model, you show there's a variable Q. So what's that Q? Is that an input, the Q concentration, or?
Is this what you're talking about here, these Qs?
Yeah.
Yeah. So I'm an engineer, right? So everything to me is a reactor. So Qs are volumetric flow rates. So this is the flow rate of blood into the muscle and the flow rate of blood out of the muscle, and it carries with it whatever's bound to the blood, right?
Okay. So Q. So what other inputs? So you have the Q, the volumetric flow rate, and also the concentration in and out.
Right. So if it was just flow, so if you have what's called a flow limited model, it's just the concentration times the flow rate gives you the mass rate in and out, right? But with PFAS also, as it flows through the blood within a tissue, it diffuses into and out of the cell into the tissue compartment itself. So there's a passive diffusion constant that's like a permeability from the blood into there. And there's also the binding to the different proteins. So we do a protein balance where they look at what are the number of active sites that are available for the protein, what's the concentration of the protein in that particular tissue. And then from that, you get the percent of PFAS that's bound versus the percent that's free. And only the free PFAS is allowed to flow in with the blood and to diffuse across the different membranes.
Okay, so in this model, the in and out, input out is balanced. And I think in one next slide, of you show the concentration versus time. Okay, so that seems like it's a dynamic model, so the concentration over time. So
right. So yeah, so here we have inputs and outputs, but it's only at it's only a steady state model if the inputs equal the outputs, right?
Okay.
So you could do that. You would approach that if you had a constant uptake of the chemical and that didn't change, which is actually closer to where humans are because we're just being exposed to it all the time. But the situation we're modeling here is a single IV dose or a single oral dose that then is cleared out of the organs. So that is a dynamic simulation. Yeah.
So is this the model like able to predict the accumulation? So like in the case we drink water every day, and how much it will accumulate in our body over the time.
Yes. So the only difference that would be here. So here we only have a single input of chemical at the first time point. But if instead we had a function that said the chemical input is constant and there's this much from water and there's this much from food, then what you would see is the opposite trend of what you're seeing. It would go up and then level out. Yeah.
Okay. We have questions from the audience. I don't want.
Mhm.
Okay, so my next question is, so is this model like a specific for certain organ and for human or rat? If it's like for the specific rat or human or organ, can it model be applied to other systems, other organs?
Yes, this is the generalizability question, right? So in order for you to model a different tissue, you would have to understand what's the difference. So specifically here, we have, for example, we have blood, muscle, adipose, kidney, and everything, liver and gut, and everything else is in the rest of body. So if you wanted to model the brain, you would have to add a brain compartment and parameterize what's important about the brain and how does the brain take up chemicals. And that's something actually we're working on because we want to include the brain in our next iteration of the model. In terms of going between humans and rats, there are a lot of scaling functions already available to understand, like if you have results for a rat, what would it look like in a human, just because the rat is a very common model. But if you wanted to do like a bird, you would have to do a new model because they have very different compartments.
Okay, yeah. All right, so my last question. So this is the model involves so many like compounds. The PFAS, we have 1,000 PFAS compounds, and the system, our human system, or any living systems is you have so many proteins and so complicated, right? And you want to develop for this model, you need parameters, partition coefficients for each of this. Then, so that's involved lots of work. And nowadays, I think one word which is everyone knows about AI, right?
Yes.
So do you think that will play a role in your research?
Definitely. So already this student, Cheng, he also worked a little bit on artificial intelligence, so machine learning models to try to predict PFAS. So one of the things that we're working on is, this approach is what I call the Lego approach, because it wants to distill the information of the different tissues down to, tissues of this type have this distribution of these different components. And so reduce the complexity so that you can just say, every brain is 70% this and 30% this and 20% that. And therefore, you're able to make a little bit better broad predictions across many different compounds. And this is also why we use molecular modeling to model many PFAS for a single protein, so that what we eventually want to get is what's called a structure-activity relationship, right? We want to be able to say, if your PFAS looks like this, we can predict it's going to behave like that, which we don't have yet for these compounds, but we're getting closer.
All right. Very exciting. So we have 2 minutes left. Do we have any questions? If not, I would ask, thank our speaker again, Dr. Ng. Thank you very much for this very exciting talk.
Yeah, thank you very much for the invitation.
Yep. Thank you.
Good night.
And
thanks.
Thank everyone for joining the lecture, and have a great evening. Bye-bye.
Bye-bye.
Ranghang, great job. This is Ed. Thank you very much.
Thanks, Ed.
Thank you. I really appreciate it. Good job. I was just, I'm exhausted. I got into bed last night at 1 a.m. and was up at work by 8. So I was glad to be here, but I was also, I'm not sure I could have been fully coherent